Today we are speaking to Erwin van Wijk. Erwin is an associate professor at Radboud University Medical Center in Nijmegen, the Netherlands. Erwin specializes in the genetic background of eye diseases, including Ushersyndroom. And in fact, he's helping to develop the very first genetic therapy against Usher type 2A that is now being tested in patients. Hi, Erwin, welcome.
Thank you.
Can you tell us a little bit about that therapy?
Yes, in general, patients with Usher syndrome have small errors in their coding sequence.
Their DNA.
In their DNA. So that's being translated in functional horses of the cell, proteins. And due to these errors, photoreceptors do not function and eventually die. And therefore people will become blind.
Photoreceptors, they are the cells in the eye.
Yes, these are the light-sensitive cells in the eye that convert light into small electrical signals that are being transmitted to our brain, and therefore we can see.
Right, and this goes wrong in Usher patients.
Exactly.
Right. So how does this therapy work exactly?
So we generate small patches of genetic material, RNA. And these small patches cover up mutations that are the underlying cause of Usher syndrome in patients. And with that, we basically fool our light-sensitive cells in the retina so that these parts that are not working and contain these mutations are not ending up in the part of the DNA that is being translated in these functional horses.
So you get a functional protein even though a part is missing.
Exactly.
Because normally in Usher patients, no protein is produced?
Most of the time, no protein is being produced. So with this therapy, we slightly shorten the protein, but this protein has still residual function.
And does this work? Have you tested it?
So we use the zebrafish as a functional model for that.
Right.
So we gave this fish Usher syndrome by introducing a variant that is also seen in patient. And this fish is indeed visually impaired. So we added these genetic patches to these fish. And what we saw is that protein production was indeed restored. And zebrafish, whereas in our Usher model, did not see correctly and were visually impaired. And after treatment with our genetic patches, these fish could see again.
Right. Okay. Fantastic. But what about people?
So that's the next level, of course. So after getting these results in fish, we designed these genetic patches. And we teamed up with a Dutch biotech company and, generated the whole procedure and made all procedure safety studies, etc. And ended up being able to start clinical trials. And these were phase 1-2 trials. Phase 1 means safety in man.
Right.
Phase 2 is the very initial proof of concept that it indeed works in a very small group of patients.
Right.
And the molecule appeared to be safe in all treated individuals. And also in all individuals that were treated, all different parameters for visual function that we have assessed showed even a slight improve in vision. And after that, a stabilization of the eyesight.
Right, okay. But then, of course, you still need phase 3 testing in larger groups of patients?
Yes, that's true. After that, the first initial results were very positive, but then there was a small problem in the whole pipeline because the Dutch biotech company that we teamed up with ended up in financial trouble.
Aha.
So they had to stop all their endeavors in making this into a treatment for patients. So luckily for us and mainly for the patients, it was taken over by a French biotech company. And they recently launched a phase 2B trial. So that's an extended version of the small phase 2 trial. And that's called Luna. And after that, if the results are still positive, which we obviously hope for, then a phase 3 needs to be designed. But this is a challenge.
Right. Why is phase 3 a challenge in Usher syndrome?
Well, we are dealing with very small groups of people. So it's a rare condition. Our genetic patch treatment is only suited for people that have variants in this very specific region that we patch with our genetic patch. So it's not very common, these variants. So small groups of people, that's a challenge one. Second challenge is if you have patients that you compare to each other, which is required in a phase 3 trial, then you are basically comparing apples with oranges.
Right, because there are very large individual differences between people.
Exactly. So what we would rather love to do is to treat one eye at first and use the other one as a control, because variation between two eyes of one individual is way smaller than comparing with two different individuals.
Right, although ideally you would like to treat both eyes, right?
In the end, obviously we will treat them in both eyes. It's just for the initial phase to show therapeutic efficacy.
In fact, how do you administer a patch like that? How do you get it into the eye in the right place?
Yeah, that's a very good question. Patches are being delivered by an injection in the eyeball. And trained ophthalmologists do these type of injections on a regular basis. Right. Also not per se with these genetic patches, but with other substances for other ocular disorders. And that's a routine of care.
Right, okay. And so you've been able to establish that it ends up in the right place in the retina?
Yes, it ends up in every single cell of the retina and acts where it should do its trick.
Right. So how long will it take for this treatment to reach the clinic? 10 years?
Well, I think that it will be faster, at least depending on the outcome of the trials, of course. Generally, it takes up to 10, 15 years before a treatment ends up hopefully being on the market. We have already completed phase 1 and 2A. So I think that we will reach the market, if everything holds up, earlier than 10 years.
Right, okay. So that would be a treatment for everybody with Usher type 2A?
No, it's just as mentioned, it is a region-specific treatment for individuals having genetic variants in the region that we cover up with our genetic patch.
Right, okay. So what is your hope or your expectation for 25 years from now?
That's still a long time ahead, of course. What we are developing is other genetic patches for other groups of patients, not only Usher 2A, but also Ushersyndroom type 2C and 1F. That's what we are working on. And we are developing these further, and hopefully they will reach the market sooner than 25 years from now. Another thing is that we recently started a novel project working on regeneration of our retina and maybe also of the inner ear.
Regeneration, so that you will restore the function after it's been lost.
Exactly. So bringing back lost sight and maybe lost hearing even. And we use the zebrafish as a model because that is a fantastic creature that still has the ability to regenerate itself. So to bring back lost cells. And that's something what we cannot at the moment. Yeah, right. So we hope to learn from them and translate these ideas and this information into a novel, innovative treatment in the future.
Thank you very much for your time, for this interview, and good luck with your research.
Thank you very much.